Let's talk anaerobic cellular respiration. So anaerobic meaning we have limited or no oxygen supply. And the very first step is going to be taking a look at glucose. Okay, there's a glucose molecule. The problem with glucose, after all that effort in photosynthesis to make it, is that it's too complex to use on its own. We have to break it down. We have to break it down in a process called glycolysis. Remember, lysis meaning to cut. So let's illustrate things this way. So our glucose, C6H12O6, C6, 6 carbons, is going to undergo glycolysis. It's going to be split, and the first step is the first step of glycolysis requires an energy investment. Okay, so we are going to require the help of ATP, one there, one there. Now, kind of like the Calvin cycle, we really want the energy that's held between the second and third phosphate, and we want the phosphate group itself. So what they're going to do is they're going to drop off their phosphate. They're going to get reduced to ADP because they only have 2 now. And here we go. So that's going to split apart, and we have 2 3-carbon compounds, and this is actually glyceraldehyde 3-phosphate, the same glyceraldehyde 3-phosphate that we talked about in the Calvin cycle. Now, what I haven't shown is that attached to this, when we make glyceraldehyde 3-phosphate, there's actually another phosphate attached. Okay, so this was our first step. We had an energy investment. We used two ATP molecules to do this. In the second half, we actually require 2 ADP molecules. ADP, right? Diphosphates. That means they're ready to make a bond, they're ready to take one, and that's exactly what they're going to do. This one is going to take this one is going to take this one, and just pull those off. To help with the reaction, we have NAD+. NAD+ meaning that it is ready to pick up electrons, it is ready to form a bond, and it'll help, clean up the reaction. So we finally pull those off, and now we have pyruvate. We have a 3-carbon pyruvate. Of course, when this happens, NAD+ is going to become NADH, and those ADP are going to become ATP. Okay, so that was all for one glyceraldehyde 3-phosphate. Remember that when we attach these phosphates, it becomes unstable. It's going to split apart, and we have 2 glyceraldehyde 3-phosphates. So for this one, it's the exact same thing that's going to happen. We're going to require the help of 2 ADP and an NAD+. So 2 pyruvate molecules. Lovely, fantastic. So for our general equation, we have C6H12O6, or our glucose, plus 2 ATP, our energy invested, plus 2 NAD+. Those are our electron carriers, and that's going to form 2 pyruvate, 4 ATP, 2 NADH, and 2 hydrogen protons. You don't need to worry about the hydrogen protons too much. What's important here is that we invested 2, we produced 4. So our net gain in energy is 2 ATP. Not super efficient, but that's what it's going to do. So the next step that's going to take place in the cytoplasm, let's say for a plant, is going to be fermentation. In plants, it's called alcohol fermentation. We are going to make an alcohol group. So the general equation for this is going to be pyruvate plus NADH is going to make ethanol. Same ethanol in our wine and beers. Ethanol plus NAD plus and CO2. So where do those come from? Pyruvate, we know it comes from glycolysis. So let's draw our 3-carbon pyruvate, right? 1, 2, 3 carbons. And it's going to have a double bonded oxygen there. So that's our pyruvate. Now the first thing is this. We're going to have a carbon dioxide produced. We're going to remove a carbon dioxide. So CO2, you'll see that there's a C2 oxygens, and that is what is going to be cut off. So our first intermediate is actually just going to be what's left over. This is our pyruvate. This is our acetyl aldehyde. And this CO2 is going to get cut off and, diffuse out. This process is called decarboxylation. The CO2 group, we call it a carboxyl group, so because we're removing it, we're decarboxylating the molecule. Great, fantastic. We made a CO2, and now we required NADH for fermentation. Lucky, we just happened to make NADH during glycolysis. So we're going to have an NADH come in. Boop. NADH. And this hydrogen is going to come here and attach onto the oxygen to make an alcohol group. So our final product is our 2-carbon alcohol, ethanol. Bam! Once the NADH drops off the hydrogen, it's going to turn back into NAD+. But the question is, what about energy? Did we produce any energy? Did we make any ATP up here? Did we release any energy? No, not really. Then why ferment? What's the point of fermentation? So if you look at the start of, the second stage of glycolysis over here, see how we required an NAD+? Guess where that NAD+ comes from? Under anaerobic conditions where we have limited oxygen, our NAD+ is actually regenerated by fermentation. This NADH that we make is actually the NADH that comes up over here. Blow. That drives our fermentation process. So it's not very efficient. We're only really making a net 2 ATP per cycle per glucose. But if you don't have oxygen, this is the best you're going to get. Okay. In animals, it's a slightly simpler process. We undergo lactic acid fermentation. So our general equation this time is going to be pyruvate plus NADH and hydrogen. You don't need to worry about that too much. Lactic acid and NAD+. So I pyruvate again, 3-carbon molecule, just like before. This NADH, like in alcohol fermentation, this NADH is the one that's coming out of glycolysis. And it's going to mechanically do the same thing. It's going to come by, it's going to have NADH, it's going to drop off that hydrogen, it's going to turn into NAD+ after it drops it off, and it's just going to turn that into an alcohol group on the side. The rest of it actually stays the same. So this was our pyruvate, and this is our lactic acid. Same thing as before, we are not making any energy. We are not making any ATP or anything like that, but we are regenerating NAD+. Like before, this NAD+ is going to come over here into the second stage of glycolysis to help turn the glyceraldehyde 3-phosphate into pyruvate, and the NADH that's produced is going to be the one that drives this reaction. So in animals, you don't have enough oxygen. Let's say you're doing some very, very strenuous exercise, you don't have enough oxygen. You're gonna you break down your glucose, undergo lactic acid fermentation because you don't have enough oxygen, and then make 2 ATP and just go through the cycle. Not very efficient, but it gets the job done. If we did have oxygen, then our pyruvate would actually enter the mitochondria into the aerobic cycle, and we will talk about that next time. Really quickly though, so these general equations, that's for one pyruvate. How many pyruvates did we have entering this? 2. One glucose is going to be split into 2 pyruvates. So, I don't know. This is going to happen twice. This is going to happen twice. Okay, so per one glucose molecule that undergoes alcohol fermentation, we're producing 2 ethanol molecules, 2 NAD+ molecules, 2 CO2s, etc.